So sorry. Other specialized waxy substances like mycolic acid, okay, that can be present in this cell wall. Okay, so specifically when we talk about your bacteria, you must have learned that depending upon what the cell wall is made up of, you have your gram positive bacteria and your gram negative bacteria. Okay, so gram negative and gram positive, from where these names come from, or from where this differentiation comes from. You have a particular type of staining that is called as your gram staining. Okay, gram staining is one of the like it is a way of basic characterization of your bacteria. Okay, so the first thing that you want to find out whether if you have isolated any organism, you need to find out, or you want to identify that particular organism. The first thing that you need to do is you have to find out whether that organism is gram positive in nature or it is in gram negative in nature. Just quickly in the chat box, can you people tell me that in case of gram positive ones, how the cells look like? What is the color of the cells that it will have? In case of gram positive cells, what is the color of the cells? Yes, it is purple. Yes. And in case of gram negative ones, yes, gram negative will look pink. Very good. Okay, so you already know what is the fundamental of gram staining. Okay, so I'm not going to go in that more of details. So here we are not going to do any differentiation as such. Okay, we are not going for gram staining, but here we want to see the cell wall structures. Okay, so the cell wall structures can be stained. So here specifically the cytoplasm has to be given a different color or has to be colorless, or and your cell wall has to be given some other colors. So there are differential staining techniques which are, or different staining techniques which are present. You have Chances method, you have Ringer's method, you have Dyer's method. Okay, so Chances method is the one which is specifically used, and apart from Chances method, the other method which is preferred for cell wall staining is your Dyer's method. Okay, so today we will be seeing in detail uh, how your Chances method and your Dyer's method is going to be done. Okay, so what is the specific requirements of those things, or what requirements, as well as what is the principle of this particular method. So all these things we will be seeing in this particular lecture. Okay, so let us go forward with the first method, that is your Chances method. So the aim of this experiment is to stain the bacterial cell wall by Chances method, as I have already specified. The requirements of this particular experiment is you require a clean grease-free slide. I'll also show you how to make grease-free. Why you say grease-free is, see, all these cells, whatever you, or organelles, whatever you are staining, you will look under oil immersion lens. Do you people know what are oil immersion lens? Yes, quickly in the chat box, yes or no? Oil immersion lenses? Yes, students? Yes. Okay, very good. So you already know what it is. So here, instead of a cover slip, you would be adding a drop of oil. And then the oil immersion objective has to be dipped in that, or it has to, so you have a kind of an sandwich that will be happening. Okay, so let me just explain you that. So I'll have say this as my slide. So on this slide, I'll have this placing of your one drop of whatever the oil is there, and then when the objective comes, okay, objective of the microscope is there like this. So the objective lens will be just touching on this oil. So here, here is this smear. That means whatever the organisms are there that would be present in this area. On that you are putting that oil. So there is a connection. Okay, so the objective is so very close to your slide that it is literally kind of touching the slide, and in between what you have, so you have this slide, you have the objective, and the objective should be dipped or it should be in contact with this drop of oil which is there. Okay, so that is how your oil objective lenses or oil immersion lenses are going to work for. So what happens is, when you, so the same slides you are going to use for many times. So the second time when you have used already oil immersion, so the your slide becomes very greasy. So it is very much important. So when you are making a smear, I'll show you a video of making smear and all those things also. So next time when we are making a smear, what will happen? You will not get a uniform smear, or the organism is not going to be uniformly present on your slide. And then you will not get a good slide, or you will not be able to, you will be able to see only blotches or patches under the microscope. Okay, so a good staining will not be done in that case. So a very important requirement is a clean grease-free slide. So what you do is you will clean it with the detergent. Whatever your slides are there, it has to be properly cleaned with the detergent, and then just 1 or 2 times you have to move it from the flame. Okay, so flame के ऊपर से निकाल देंगे. So what happens is all the oil, if it has not been removed by the detergent, definitely by the heat, it would be definitely removed. And then you just wipe it off with a clean cloth, not with cotton or tissue papers, because then you can have fibers of this cotton and tissue papers on the slide, and those will also be stained, and then you may get confused between those fibers and the microorganisms. So these are few basic things that you need to remember whenever you are doing any staining procedures. Okay, so staining is one of the very, very important parts of your biotechnology or microbiology, whatever you are doing, or cell biology. Okay, so fresh cell suspension is being required. So what is cell suspension? You have these microorganisms that would be taken, or the colonies are going to be taken into saline suspension. So saline is an isotonic solution. Isotonic solution in the sense it will have the same amount of salt, okay, same amount of salt as it is present inside the cells. So that, so what happens in case of your hypotonic solution? So if it is an hypo means what? Less salt, okay? So less salt. So if you say you have this, say a beaker, and you have this hypertonic solution in this. Students, can you see the annotations that I'm making on this slide? Just speak out. I'm not able to see your chat because I've kept it full screen. I'm not able to see your chat. Just someone just unmute and tell me, can you see the annotations that I'm drawing onto these slides?
Yes, ma'am.
You can see these things in red which I'm drawing?
Yes, ma'am.
Okay, okay. Thank you so much. Okay, so see, I have two beakers. Say, for example, so in this I have hypotonic solution. Hypo, that means the salt is less in the solution which is there over here, and this is hypertonic solution. Okay, so that means the salt is more in this particular solution. Okay, this is hyper in nature. So salt is less. Salt is less. That means what? The water is more. Please understand this. Salt is less. That means water, H2O, is more. है ना? And here salt is more. That means H2O is less. Okay. Here, when I say H2O is less, or H2O is more, or salt is more, or salt is less, I am talking about in terms of what is the concentration inside the cell and outside the cell. Okay, so that is what we are referring to. So if your cells are, so now let me just take another pen color and show you here. So say for example you have these cells which are suspended over here. So now what happens is the water concentration is more outside this cell as compared to inside this cell. Okay, so there is a pressure which is built up, which is called as your osmotic pressure. Okay, so there is a pressure which is built up on this cell wall, and water will always move from a higher concentration to lower concentration, right? So water is more outside. So what will happen now? Water will start moving inside this cell. What will happen if the water moves inside this cell? The cell will swell up. Okay, the cell will swell up. It will take the water inside and it will get swelled up. And when it gets swelled up, there is a limit to it. After some time, the cell will burst out and the cells would be killed. Okay, so that is the condition when your hypotonic solutions are used. Hypertonic में क्या हो जाएगा? So water concentration is less outside and it is more inside, right? So again, the water is going to move from inside to outside because concentration of water is more inside the cell and less outside the cell. So water will move from inside to outside. So in this condition, this cell will shrink. What will happen to this cell? It will get shrink. Yes. See, these are nothing but these are just the basics of staining or basics of why we are using saline suspensions and all these things that I'm trying to make you understand. So it will shrink and again finally the cell will die. So to avoid all these things, we should have a saline suspension, okay, where it is isotonic. Iso means same. Iso means same. So it should be an isotonic solution where the concentration of salt is same as inside the cell and outside the cell. So specifically, it has to be 0.85 to 0.9%. Okay, so that is what is expected. And that is what your cell suspension will be made up of. Yes, a quick yes or no. Have you understood why an isotonic solution or what is a cell suspension? Yes, students? Okay. Great. So next thing, what we require is 0.5% basic-Fuchsing solution, okay, which is a dye, and you have 0.5% of Congo red solution, which is again another dye. Okay, so let us see the mechanism or the principle of cell wall staining. So you have this smear is air dried and very, very important. Okay, so this smear will not be heat fixed. Okay, usually whenever we are making any smear, we just move it on the flame 1 or 2, 3 times so that these cells get fixed onto the your slide. Okay, so when you are giving a wash, the cells should not get washed away or from the slide. So it is very much needed that you fix them on the slide. Okay, but in case of your cell wall staining, this heat fixing is not to be done. Okay, why? Because when you heat fix it, it will alter the structure of the cell. So that is very, very important thing over here. So here the slide has to be only air dried and stained. So as we know, cell wall and cytoplasm are acidic in nature, where cell wall is more acidic than the cytoplasm. Okay, so that is the reason why we will be using basic Fuchsin. From the name itself, you understand that it is basic in nature. So because your cell wall and cytoplasm is acidic in nature, when you are adding the first stain, that is basic Fuchsin, it would be taken up by both your cell wall as your cytoplasm. Yes, but what I told you, that it is very much necessary that the color of the cytoplasm and the cell wall should be different. Then only you would be able to distinguish the cell wall from the cytoplasm. Am I right? So अभी यहाँ पे first stain देने के बाद हमने क्या देखा? Both of them are going to take the same color. So that is not going to serve our purpose. So, but here we want to stain the cell wall, as I said. So the smear is treated with the second stain. So a secondary stain is being used, that is your Congo red. So Congo red is also acts as a selective decolorizer. Even if it is a stain, when it is used as a secondary stain, okay, so here it is a secondary stain. Which was your primary stain? Basic Fuchsin. So your Congo red is acting as your secondary stain. So when it acts as a secondary stain, acts as a selective decolorizer. So what it will decolorize? Only the acidic portion of the cytoplasm, and the cell wall will remain as it is. Okay. So what is happening? First, your cell wall and your cytoplasm both took up this basic fuchsin, which is again pink in color. Okay. So both have taken that. Then you add your secondary stain, that is your Congo red. But Congo red क्या करेगा? It will only decolorize your cytoplasm. So whatever your red or pink color is there, so that color is going to be removed from the cytoplasm. But the cell wall will remain as it is. And hence, you can see the cell wall stained with your basic fuchsin. So this is the principle of cell wall staining. Yes, students, quickly in the chat box, have you understood or not? If anyone wants me to repeat this, kindly tell me. Have you understood the principle of the staining procedure? Okay, okay, good. So let us move forward. What is the procedure? Okay, how do we do the procedure? Simple. Take a clean grease-free slide. Make a smear with the help of sterile loop. Okay, I'll show you a video of how to make the smear and all those things. Okay, so you give this sterile loop, you air dry, and do not heat fix. Okay, the video that I will show there, I will show you how to do the heat fixing also. But please remember, in case of cell wall, you don't have to do the heat fixing. So the video that I will show, it is a general video for monochrome staining, like how this staining is being done for all the staining procedures. It is a common practice. Okay. So once the slide is dried up, you have to flood this smear wherever your cells you have made. So there you have to put basic fuchsin solution for 3 minutes. After 3 minutes, you remove whatever the excess stain is there, and you don't have to give any wash over here. So on that only, you will add the Congo red solution for 4 minutes. And after 4 minutes, then you will just give a gentle wash and the side with the water. So once the washing is being done, you will again air dry it, because you have to put oil in that, right? So for putting the oil, it is very much important that the slide is again air dried, and then you can observe under the oil immersion lens. So when you observe under the oil immersion lens, you can see this kind of a picture. Okay, exactly same. This what is how you are going to see under the microscope. You can see over here red color cell wall, and the cell, whatever your cytoplasm is there, that is colorless. Okay, so usually the organism that we can use different types of organisms. So this is the organism of Micrococcus species. Okay, Micrococcus lutea, which gives you a beautiful yellow colored colonies. Usually that particular organism we use for cell wall staining. If you are using a bacillus, it will give you dot shaped cell walls. Okay, so depending upon what is the structure of your bacteria, you can see that the cell wall structure would be that way. Okay, so you can see over here, this is how you write the observation also, students. Please understand, when you start writing your journals and all, you have to write it like this. So under your oil immersion lenses, you will see how your colorless cytoplasm and pink colored cell wall of the bacterial cell wall can be seen. Okay, so that is about your chances method. So I'll just show you the basic how to do the staining thing. Okay, just give me 1 second. plate where we have different types of culture. So let me just show you this plate. You can see there are different colonies which have been growing of different colors, different sizes and different shapes. Okay, so here we have isolated the cells or the organisms which are there in the soil. Okay, so now if you want to characterize them, we need. Students, you are able to see the video, right? I've just paused it. Just let me know you are able to see.
Yes, ma'am.
And here?
Yeah, ma'am.
Yes, ma'am.
Need to study them. So the first step is going to be the monochrome staining to just see what is the shape of the whatever the organism is there. So today I'll be showing you how to do it for one of the cultures. Okay, so I'll be choosing this yellow one. So yellow colony that you are able to see over here, I'll be choosing that one. This is called as a nichrome loop. Okay, so this is an inoculating loop. This is the equipment that we use to transfer any of our cultures directly from plates to either suspension or on slides. Okay, so first I'll be telling you how to work aseptically also. When I say aseptically, it means that between these two burners. So when we light the burner, the temperature around that area increases. So all the organisms which are there in that nearby environment, they would be killed, and you would be able to study the organism of your interest only. So let me just light this burners. So both the burners, the distance should be. This is how we have to work between the burners. So before inoculating, we have to make sure that the inoculating loop, that is the nichrome loop, is sterilized. So how do you sterilize it? The entire loop, that is this particular portion, should be red golden. Okay, red hot. So why nichrome loop is being used? Because this is one of the characteristics of nichrome, that it gets red hot very fast and then cools down also very fast. So then you have to cool it for some time. Then only you can touch the microbe or the colony with this loop. Okay. So I am just holding it between the burners. So during this time you are not supposed to take out the loop from the burners. Okay, you have to keep it in between because this is the area which is going to be sterilized. One more rule that you are not supposed to speak. Okay. So now I will open this plate, and we have to take care that we are not opening the plates with the microbes without this burners. Okay, you have to open it only between the burners, never open it outside the burners. Okay, so I'll just take some of the culture in my loop, and then this is saline. Okay, normal saline. So I'll mix it, and this will make my cell suspension. So again you have to sterilize your loop so that you are killing all the organisms on this loop, and then you can keep it. So you can see the loop turning red hot. Okay, again cool it down and then keep it aside. So now I'll just mix this culture in this saline. So this will give me a saline suspension. Okay, so this saline was also sterilized. It was autoclaved. So this kept with this form that is there. That means it is sterilized. All the other organisms. So now I will be showing you how to make a smear on the glass slide. So you can see here that I have made a marking. This marking is around 1 centimeter by 1 centimeter square. So we have to make this smear inside that marking, inside that area. Okay. So make sure that you can see that I have made the marking on this side. So the smear that I will be making would be on the opposite side. Okay. So again we will take the nichrome loop. We are going to sterilize it. So just see how it turns red hot again. Okay, so just keep it for 5 seconds. Okay, and then let it cool down. Once it has cooled down, then we will be taking our suspension. So just your loop in the suspension, you can see here there is a bubble onto this loop. Let's see. Yes. So when this bubble is there, that means the culture is already on the loop. Okay. So now you can just take the slide and then fully rub onto this entire area. Okay. You can take the culture only once, or you can also take it twice. And evenly spread it in that entire area. Again, sterilize the loop, keep it aside, and let this slide dry up. Okay, first we need to air dry the slide, and then after that is done, we will heat fix it. The slide has already been air dried, and we will be doing the heat fixing of this slide. Okay, so why do we do the heat fixing? So that when we are pouring the dye and we are giving the washing, the organism should not be washed away. So it should be fixed properly on the glass slide. So just 1 or 2 times we have to just allow the slide to move on to the flame. Okay, so our heat fixing is being done, and now we will proceed for staining. We continue with monochrome staining. Our smear is ready now. So you can see this is the staining stand on which we can keep our slide, so that whatever the dye is there, that will fall on the basin and you are not staining yourself. You have to take care you don't stain your clothes, you don't stain your hands. Okay, so in the area where we have made the stain, we will just add 2-3 drops of this stain. This is crystal violet, and we will incubate it, or we will keep it for 1 minute. So 1 minute is done. Now we will just decant. Okay, and slowly under running water, we will allow this dye to be washed off. Okay, so once it is done, again you have to air dry this slide. Okay, that the slide has air dried. So air drying is very important, because here we are not going to put any cover slip. We are supposed to see it under compound microscope under 100x. So for 100x, we have to use this cedar wood oil. This is cedar wood oil, and just a drop of it. Using this capillary, I will place just a drop of it at the center of my smear. Okay, not much, just enough, and then you have to place it on the stage. So this is a compound microscope. So the objective has to touch. It has to touch the slide, and the adjustment should be such that you have the slide, you have the objective, and it should be connected by the oil. This is the coarse adjustment. This is the fine adjustment. So slowly you have to come up using your coarse adjustment by looking under it. So this is the mirror which will allow the light to be focused on the objective lens. You can adjust it using this.
So once you have done the coarse adjustment, you can adjust it using the fine adjustment to see it properly. So this is the eyepiece, okay? So this is 10x eyepiece that you can see. So objective is 100x and the eyepiece is 10x. So that is how 1000 times you are magnifying the organism and you can see it. So this thing, like this is the stage where you can move it back and forth. You can move it left and right, and that is how you can observe the slide. So now what I can see is, because I had used crystal violet, I can see violet color. This is the organism that I have used is Micrococcus. Hence, we can see clusters of small cocci, purple colored or violet colored cocci under the microscope. Hello, everyone.
आपकी व्यूज ज्यादा नहीं है।
मत मिलने दे, तो डालते तो रख, नहीं मिलता है, नहीं मिलता है, अलग बात।
Okay, students, have you understood this? So that was a basic video that I have created for you people to understand how the staining is being done and the basics of it. So as I told you, that particular video was for monochrome staining, right? But today what I have explained is about your cell wall staining. So that is the basic one. So basic for every staining procedure, those things will apply, right? I will also share the link for that particular video. If at all you want to see that video again, you can just go through it, okay? For any type of your queries. I have tried to explain it as in depth as possible, okay? So that was for your cell wall chances method. Let us now see for Dyer's method. So here again, what you require is your young culture of bacterial cells. So that is your cell suspension. So here you require a different, instead of basic fuchsin, you have a different state. That is cetral peridinium chloride solution. And then you have Congo red and you have your methylene blue. What is the principle? So this particular solution, that is your cetral peridinium chloride, is nothing but it is a quaternary ammonium compound. So this compound, whenever it gets mixed with water, it will form this positively charged cetral peridinium ions and negatively charged chloride ions. And the cell wall of bacterial cell is negatively charged. We have already seen that. And when this positively charged cetyl peridinium ion comes in contact with the cell wall, they get attached to the negatively charged cell wall and make the surface positively charged. So further, when the smear is treated with Congo red, which is an acidic dye, then the cell wall gets that red color, whereas the cytoplasm will be stained with the methylene blue and the cytoplasm will get the blue color. So let me just explain again quickly. You have this cetyl peridinium chloride. So when it is in water, it will give rise to positively charged ions, that is your cetral peridinium ions, which will go and bind to only the cell wall. Okay, because the cell wall is exposed. Okay, cytoplasm isn't exposed. So it will bind to the cell wall because cell wall is negative, more negative in nature as compared to your cytoplasm. So what will happen is it will give, so it will make the cell wall positively charged. And then Congo red, which is acidic in nature, that means negatively charged, that will go and bind to the cell wall, but it will not go and bind to the cytoplasm because cytoplasm is negatively. Oh, okay. Sorry, I just forgot. Okay. So what will happen is it will not stain. It will not stain the cytoplasm. The Congo red will specifically, selectively stain only the cell wall, but not the cytoplasm, right? And only the methylene blue, that is the secondary stain that we are using, that will stain the cytoplasm. Okay. So procedure is again the same. You will, so here you can heat fix this smear. It is you can or you may not. Then add 3 to 4 drops of cetyl peridinium chloride. Now 1 drop of Congo red on cetylpyridinium chloride and mix them, rotating the slide slowly. So the slide has to be kept for 2 minutes. Water wash the slide and counter stain the methylene blue and keep the slide for 10 seconds. Okay, and wash the slide with water and observe them under oil immersion. So if you don't want to stain the cell, like in case of your chances method, you saw that the cell wall, the cytoplasm was colorless in nature. So if you don't want it, so you can just avoid using methylene blue. So if you're not adding methylene blue, your cytoplasm will not be stained, and you will see a similar kind of a picture as in case of your whatever your chances method was there. Okay, so this is just the same procedure. So this is how you will see the cells. You can see over here red colored. So this is a bacillus. So you can see the red colored cell wall and blue colored cytoplasm, okay, which is a little different. So I will show you another simulation link for, these are present online. Where you can see how these type of your just give me 1 second. Let me show you that tab. So these are simulations which are present, or you can have virtual labs present on internet. Okay. So can everyone see? Can everyone see this virtual lab? Everyone?
Yes, ma'am.
Yes,
Okay,
ma'am.
okay. So you can see how the simulation can work. So step by step, it will show you. So let us start. So it will show, like, take the first slide. So you take the slide. Okay. So here we are washing the slide first. Okay. So washing it and making it grease free. You can see how we can do it. So you wash it, take detergent, wash it with detergent properly. Yes, again give a wash. Okay. Then take the slide. I told you, just move it 2-3 times on the burner, so that makes it grease free. Then you take the cell suspension. This is your nichrome loop. I showed you in the video also, right? So nichrome loop, you dip it, take the culture, and then smear it. Make it a smear. Okay, so you can see. Then let's start with the staining. So you can take the first cetylpyridinium chloride. Yes. Then you will take the Congo red. Okay, keep it for 3 minutes. You give a wash. So whatever excess stains are there, that is going to be removed. Then take methylene blue. Again, wash it. And then we will mount onto your microscope. Okay, so then you can observe and you will see that kind of your picture which I have already shown it to you. Am I clear, students, everyone?
Yes, ma'am.
Yes, ma'am.
Yes. Is it very much clear? Have you understood this? This was your first staining method, right? For your bacterial cells. Okay. So I have gone in detail about each and everything. Okay. So now there are many staining procedures which are there, which would be taken by Navami ma'am. So then ma'am will take it a little bit faster, because now you know what is the basics of staining. So she will just explain you about the principle of heat staining method and then how to go about it. Okay, so the basics will remain the same, only the stains will differ. Okay, and procedure may differ here and there a little bit depending upon what is the organelle which you need to stain for. Am I clear, students, everyone? Okay. Okay, fine. I will share this recording also in the classroom.